Hi friends, welcome back to Biochemistry. In this third module of gluconeogenesis, we deal with the regulation of gluconeogenesis. Now we know the significance of maintaining a normal level of blood glucose level around 70 to 110 milligrams per deciliter, and this is basically achieved by regulating basically two processes. One is glycolysis, the other is gluconeogenesis. So regulation of gluconeogenesis cannot be done independently. It is always reciprocally regulated. That is, glycolysis and gluconeogenesis are reciprocally regulated. And generally speaking, gluconeogenic enzymes are active during the period of starvation, and glycolytic enzymes are active during fed state. So let us see the regulation, reciprocal regulation in detail. The metabolic regulations are done so as to avoid futile cycling. What are futile cycles? They are a set of opposing reactions that can be arranged, that will take place in cells and tissues and in cycles or chains, but they do not usually occur simultaneously. Like glycolysis and gluconeogenesis are reciprocally regulated, which means such reciprocal regulations are done so as to avoid futile cycling of metabolites and futile cycling of energy. In fact, one of the prime aim of metabolic regulations is to achieve this, so as to avoid wasteful expenditure of energy and resources. Now, these are the key enzymes of gluconeogenesis. We have gone through this in the second module of gluconeogenesis. We can go through the second module: pyruvate carboxylase, phosphoenol pyruvate carboxylated, fructose 1,6 bisphosphatase, and glucose 6 phosphatase. Now, the regulation of these enzymes are achieved by three mechanisms. The first one is change in the rate of synthesis or production of enzymes. Now, this is basically done by controlling the enzyme production by induction or repression. Here, enzyme induction refers to the increase in the amount of enzyme produced as a result of some stimulus. Maybe by hormone or other inducers which stimulate the production of a particular enzyme. Whereas enzyme repression, it's the opposite idea, which refers to a decrease in the enzyme synthesis after a similar kind of stimulus. Now, this is one of the methods to control the availability of enzyme. We will see it in detail. Now, second one is covalent modification by reversible phosphorylation. This is the most remarkable covalent modification in enzyme regulation. Amino acids in proteins or amino acids in proteins or enzymes, the amino acid residues like serine, threonine, and tyrosine are common amino acids that participate in modification of phosphorylation, and they are used to control the enzyme's catalytic activities. In phosphorylation, a phosphate group is transferred from one molecule onto the enzyme, so thereby turning the enzyme sometimes on and sometimes off. So phosphorylation can actually turn the enzyme on in the case of some enzymes, whereas at the same time it can turn the enzyme off in the case of some other enzymes. So both activation and inhibition are possible by this phosphorylation or reversible phosphorylation. The third mechanism here is allosteric modification. Now, in biochemical reactions, allosteric regulation is the regulation of an enzyme by binding of an effector molecule, modulator molecule, at a site other than the active site of the enzyme. There will be an active site. So this particular effector or the modulator attaches at a site other than the active site of the enzyme. The site to which effector or this molecule binds is termed the allosteric site or regulatory site. And allosteric regulation of enzymes is crucial for the control of cellular metabolism, which again avoids futile cycling of materials and energy. Allosteric control permits a more dynamic and complex control of enzyme activity, obviously thereby conserving the resources of the cell. Let us see the details of regulation of gluconeogenesis one by one. Enzyme induction and repression are basically done at or controlled at hormonal levels, and as always in carbohydrate metabolism, the hormones glucagon, epinephrine and glucocorticoids are on the one side. They are generally catabolic and they favor gluconeogenesis. Now, whereas the anabolic hormone insulin is on the other side, it did not promote gluconeogenesis, but it promotes glycolysis. Here, glucagon, epinephrine and glucocorticoids, they are released as a result of low blood glucose level or during prolonged starvation. And actually both epinephrine and norepinephrine, both of them increase hepatic glucose output through gluconeogenesis. And glucagon here induces gluconeogenic enzymes, namely pyruvate carboxylase and phosphoenol pyruvate carboxylated. On the other hand, insulin actually exerts direct control on gluconeogenesis by acting on liver, as well as it affects gluconeogenesis by acting on other tissues also, so that gluconeogenic substrates are not made available. So that is one strategy of insulin controlling gluconeogenesis. Insulin, which means it represses gluconeogenic enzymes. In fact, the high glucagon insulin ratio, it always favors gluconeogenesis. Now, covalent modification. Covalent modification is achieved, as we had discussed, through reversible phosphorylation. Now, one thing we have to remember here is that the key enzymes of gluconeogenesis are active in the phosphorylated state. So, this is the point, that when the tissue is having a general environment of phosphorylation, it favors mobilization of glucose through gluconeogenesis. And this is the same in the case of glycogen breakdown also, glycogenolysis also, because it is another process which actually contributes to elevation of blood glucose level. In both cases, enzymes involved get activated by phosphorylation. I can see here, besides induction, these hormones, glucagon and epinephrine, they are also involved here in phosphorylation also through these processes. Now, actually, yes, activation of formation of cyclic AMP and the cyclic AMP dependent protein kinases. And this activation cascade has been illustrated in a previous session of our lecture, that is regulation of glycogen breakdown. You can go through it. So finally, this actually go on to stimulation of gluconeogenic enzymes. And glucagon is unique in that it also inactivates PDH. It is through a cyclic AMP mediated phosphorylation process. Now, PDH inactivation means pyruvate dehydrogenase inactivation means pyruvate will not be converted to acetyl coenzyme, which means pyruvate will be available for gluconeogenesis. So, pyruvate can be converted to oxaloacetate and can be taken to gluconeogenesis. But insulin here, it generally causes dephosphorylation of gluconeogenic enzymes in the same way that it affects the glycogen breakdown enzymes also. So, in any case, insulin is the lone fighter here and causes dephosphorylation or environment of dephosphorylation. And we go on to see allosteric regulation. While discussing allosteric regulation, the energy charge of the cell will have to be taken into consideration. Energy charge means the energy charge is ATP to AMP ratio is high, that is ATP is high. No more ATP is needed and no more glycolysis is needed and glycolysis is halted or stopped. If energy charge is low, that means ADP, AMP level is up. That also means ATP is low. That means glucose is required for production of energy or production of ATP and gluconeogenesis is promoted. So, this is the basic idea of the reciprocal regulation of glycolysis and gluconeogenesis. As well, some of the intermediate substrates also come into play in the metabolism as feedback agents during gluconeogenesis and glycolysis. We can see in detail. The first one here is glycolysis shown here. We can examine the effect of these substances, these allosteric modifiers, one by one. Now, in glycolysis, the pyruvate kinase step is shown here, the last step of glycolysis, where fructose 1,6-bisphosphate. It is one of the substrates which accumulate, if gets accumulated, it promotes glycolysis so as to produce pyruvate. Whereas acetyl coenzyme, that is the product of pyruvate, pyruvate gets converted to acetyl coenzyme. If acetyl coenzyme gets accumulated, they will have a negative feedback. And ATP also, if ATP is high, that means energy charge is high, no more glycolysis. Here, alanine, the role of alanine is important here. We know that there is an alanine pyruvate cycle. So if alanine is in plenty, that means it can get converted to pyruvate. So pyruvate accumulates. Once when pyruvate gets accumulated, no more glycolysis is needed. And pyruvate, in fact, it can be a channel to gluconeogenesis through oxaloacetate. So that is the effect of alanine. And what happens during gluconeogenesis? There are two enzymes, obviously we know, and these molecules here. ADP, if ADP is high, that means energy charge is low, as we had discussed. Gluconeogenesis is promoted. And acetyl coenzyme, accumulation of acetyl coenzyme is having a positive effect here. So that was a negative in the case of glycolysis, for obvious reasons. Now the allosteric regulation of fructose 1,6 bisphosphatase. This is actually the key enzyme or the most rate limiting enzyme of glycolysis. You can see the effect of these molecules one by one. Fructose 2,6 bisphosphate. This is actually a pure allosteric molecule which enhances the activity of phosphofructokinase first. Now AMP, ATP, this is obviously energy charge. High ATP means high energy charge, no more glycolysis is needed, as we had just mentioned. Here citrate. Citrate is actually the substrate of a TCA cycle which is produced from acetyl coenzyme, which in turn is produced from pyruvate, which means that if citrate is accumulated, it can have a feedback effect on glycolysis and have a negative feedback effect on glycolysis. And what happens here in gluconeogenesis? These molecules, the same molecules which have been mentioned here. Fructose 2,6 bisphosphate, obviously it enhances PFK, it actually have a negative effect on fructose 1,6 bisphosphatase, the enzyme of gluconeogenesis. And AMP, accumulation of AMP means we have already discussed the effect of ATP AMP ratio here. And citrates also, citrate if accumulated, it has a positive effect here because it halts the process of glycolysis and at the same time it enhances the gluconeogenic enzymes. Now, allosteric regulation of glucose 6-phosphatase, another key enzyme of glycolysis. Obviously, glucose 6-phosphate has a negative effect. It's a feedback inhibition. Glucose 6-phosphate is the product here, and glucose 6-phosphatase acts on glucose 6-phosphate to give glucose. So this is the different mechanisms by which glycolysis and gluconeogenesis are reciprocally regulated. Thank you.